Okay, so the work in our group really aims to understand both the causes and consequences of molecular variation in the brain and the role that this may play in the onset and etiology of neuropsychiatric disorders and neurodegenerative diseases like Alzheimer's disease. And as Phil Ingham referred to earlier, these are an important area to study given their increasing burden in terms of healthcare costs. So what our work has shown is that despite having a very highly complex underlying etiology, these are very complex disorders caused by a very complex interaction of genes and environments. At a molecular level in the brain, we actually see a fairly convergent molecular pathology. And we see this by measuring RNA abundance and also modifications to the epigenome in disorders including Alzheimer's disease, autism, and schizophrenia. So we take a range of approaches to annotate the human brain genome. We use a series of great brain resources that we have access to. We study discordant monozygotic twins. We apply our technology to clinical sample cohorts and are increasingly moving into the field of functional biology using animal models, cell models, new emerging genome editing techniques and single-cell profiling methods. And just to say that we take a very integrated omics approach, so we look at genetic variation, integrate this with epigenomic gene regulation and transcriptomic variation, taking high-level systems-level analyses. So just to give you a flavor of some of the work we're currently involved in, so we realize, obviously, that the genome is much more complex than a series of A's, C's, G's, and T's, and our group is involved firstly as part of the NIH Roadmap Epigenomics Consortium in mapping functional regulatory modifications sitting on top of that DNA sequence. And more recently, we've become involved in the PsycENCODE project, which is aimed at mapping functional regulatory variation across all the cells and tissue types and brain regions that obviously are involved in mediating brain and behavior function. And this is a useful approach because what we can do is take what we've learned from genetic studies of human disease and overlay this with what we know about functional regulatory variation in the genome to map regulatory elements, enhancers, and ultimately generate new hypotheses about how these genetic variants impact upon gene function. So I'll just talk you through a couple of our studies. So something we're very interested in given the neurodevelopmental origins of disorders like autism and schizophrenia is actually what is going on in the brain during normal human brain development. So we've profiled regulatory variation across a large swathe of human brain development. This top figure just shows changes, dramatic changes in DNA modifications, DNA methylation and DNA hydroxymethylation occurring across human brain development. We can see large-scale sex differences in gene regulatory networks between males and females occurring very early on during brain development. This is interesting given what we know about sex difference in the prevalence of disorders like autism. And we also are able to use this annotatory genomic data and lay this on top of genes that we know play a role in disorders like autism and actually highlight regions of these genes that are likely to be important during those key points of neurodevelopment when we know things are going wrong in the wiring of the brain that ultimately leads to disease. I'll just finish. Most important slide is this, which is the group who've helped me with this work. Thank you.